All right, fantastic. And I'm going to put a spotlight on Rahul. Fantastic. Okay, welcome everyone for the SLAAM talk today. It's my great pleasure to invite and introduce our speaker for today, Rahul Chajwa. Dr. Chajwa is a physicist exploring the principles governing self-assembly and transport in the ocean's biological pump. His research focuses on bridging biological oceanography and soft matter physics to develop predictive frameworks for ocean-based carbon sequestration. Currently, he's a postdoctoral researcher at Stanford University working in the group of Manu Prakash. He earned his PhD in physics from the Tata Institute of Fundamental Research in India under the mentorship of Sriram Ramaswamy, Narayanan Menon, and Rama Govindarajan. He worked on some very, very interesting problems at the interface of fluid dynamics, sedimentation, and non-equilibrium statistical mechanics. And currently, his transition to interdisciplinary and biological physics research in the Prakash Lab is facilitated by HFSP and their fellowships. And he will tell us some very fascinating work, I believe, today on the intersection of climate change, oceanographic biodiversity, and soft matter. And so with that, take it away, Rahul. Thank you.
Thank you, Suraj, for that kind introduction and for giving me this opportunity to present my postdoctoral work. So today I'll be, as the title says, cellular slingshot and hidden comet tails. And I hope towards the end of it, I'm able to express the meaning of these words. Okay, so starting. Okay. So my first surprise during postdoc, I joined in 2021, and in Manu Prakash's lab, and the first thing he did was take me to Monterey Bay, and we did plankton tour together. And so if you look at the ocean from the satellites, you can see this green thing waving around, which are essentially algal blooms. If you scoop some water out of the ocean and look at it under a microscope, we find this rich diversity of living things that photosynthesize. So oceans are not just mere fluid mechanical entities. They are living at some scales. And which was at that time surprising for me, for my own ignorance. But yeah, if you look at the ocean in the vertical dimension, these phytoplanktons in the upper layer of the ocean photosynthesize because that's where light is, and lower region of the ocean are nutrient rich, so they need to move around, which I'll get to. But these plankton agglomerate and sink down, effectively sequestering organic carbon from surface to deep oceans. And in a transport mechanism broadly known as the biological pump. So the clean active systems, as we know, in this canonical sense of active matter, we know the basic conceptual understanding of it has been developed over the last 2 decades, and which still continues to present exciting direction. But biological suspensions are far richer in that they have feedback, learning, adaptation, computation, especially in their ecological settings. And in this particular talk, I'll be discussing biological pump broadly and how it couples to ocean and how oceans and climate are coupled in that it sequesters carbon, a significant amount of carbon from surface to deep ocean. And 70% of Earth is ocean. So it's not surprising that it has a significant impact on our climate. Okay. Over the last 5 decades of biological oceanography have given rise to this schematic that in how a carbon is sequestered, how so marine plankton are responsible for 50% of the photosynthesis that happen on Earth. So half of the oxygen we're breathing actually comes from that. And complex biophysical interaction lead to their agglomeration then in aggregates that are called marine snow. And as it sinks through this bath of marine bacteria and viruses that decompose it, less of what is produced reaches the bottom of the ocean. And we don't exactly understand how much. So we have a good measure of how much carbon is produced by satellite measurements on the surface, how much primary production happens on the surface of the ocean, but how much of it reaches the bottom of the ocean, we do not have a good understanding of it. That sets significant uncertainties in ocean-based carbon sequestration. And the idea was the mechanistic understanding of this transport phenomena is missing. And this problem is rich in soft metaphysics, as I'll be able to present in later in this talk. So people like to say that if this pump is turned off, the carbon dioxide in the atmosphere will jump from 400 to 600 parts per million. So it is a significant knob in the climate change. Okay. So phytoplankton, as I said, are the primary driver of this sequestration mechanism. On the right, it's actually satellite data where you're seeing the chlorophyll content in the ocean. So the green region are the chlorophyll. So it's dynamically evolving, and it's in principle. So this is like a fast forward in that it's per month. It's a monthly photograph of chlorophyll in the world's ocean. In principle, it can be understood as a coupling between. So environment is that. So sunlight, nutrient, temperature, salinity, pressure at each point in the ocean are coupled to cellular behavior locally, and then cellular behavior is coupled to ecological network. I mean, it's not just one species that is present in the ocean. So this nutrient, temperature, salinity is on the fluid mechanical background in the system. And we have a good understanding of the fluid mechanics on how flows move around in the ocean. And it's a cellular behavior itself has many aspects to it. To name a few, how the cells regulate their buoyancy in the ocean, how they swim around, what is their cell cycle, how do they respond to the local environment just outside them, how do they adapt to changes in environment, which is very relevant in the context of the changing climate. And of course, how they interact with other species is also part of the cellular behavior, which has evolved in its native environment. And then there is this ecological network. So locally, there are different kinds of plankton that are there, and they interact with each other in a certain manner. So these things are not decoupled from each other in that local cellular behavior can deplete environments, for example. And the cell, for a cell or a plankton, its environment is also its ecological network. In that environment and ecological network is also not both the biotic and abiotic component that a cell responds to for the cell, it's the same. So these three, although I'm showing it as a different sets, are actually not distinct. So the idea is to be able to wrap our head around this complex system. So the one might ask, so how do we start understanding it? One way to look at it is to zoom out, just like people have done in this case, and look at the chlorophyll production. It gives you the primary protection. But the drivers of this dynamics are actually 10 to the power 12 times smaller than the planetary scale. And so we can't ignore the cellular details in order to understand this dynamics. So one might ask whether we can go from understanding cells to understanding ecosystems. But as we know, more can be different. But to make some progress, the approach we take is take a given plankton, study it in its ecological setting, in that how it would behave in the environment in which you find it, and seek general principles of that particular dynamics. So even if we can't explain, be able to describe ecosystem starting from cell, we might be able to tell how more is different in that context. Okay, so that was a long background of this talk in the outline. So I'll be talking about two two problems. One deals with how cells regulate, or a particular type of plankton is what I'll be discussing, regulate their buoyancy in the ocean. So we'll be studying cellular behavior in its ecological context, and we'll find something that is migration without swimming, and how gravity can act as an evolutionary selection pressure on these plankton. I'll be discussing, I mentioned that cells agglomerate and sequester carbon in the form of something called marine snow, which has nothing to do with ice or snow. It's essentially a biotic aggregate that has some fraction of carbon, and that organic carbon reaches the bottom of the ocean, some portion of it. So I'll be discussing morphology and dynamics of marine snow, in particular its fluid structure interaction and the role of viscoelasticity in determining how much carbon gets sequestered. And broadly, I'll end with giving like a brief highlight of ocean's biological pump and how it's related to climate change in the ongoing change. Okay, so the papers the result I'll be presenting from these two papers for those who are interested. And this work is done with, so the first part of the work is done with Manu, Adam, Hongchuan, and Manu is my PI. We also collaborated with hardcore biological oceanographers, Benjamin Van Moe and Katie Beidel from Hui and Rutgers, and with a great grad student, Ellie Flaum. And this is a scientific. So when we go in the ocean, there is this crew that we work with. And this is a photograph from the expedition I'll be discussing. And of course, a bunch of people who has helped here and there, who I'm really grateful to, and HFSP and biologists. All right. So if we collect water from the ocean at different depths and take out and look at it under a microscope, and see whether it's sinking or rising, people have found before that whether a cell rises or sinks depends on where you find it. So this is a rough profile, of course. Note that the line was fitted by eye. So this is not any type of claim here, but roughly the cells present in the upper layer. So this is particular to this result is particular to a non-swimming dinoflagellate called P. noctiluca, which is one of the many dinoflagellate that are responsible for bioluminescence in the shores. And we are not looking at bioluminescence; we are looking at buoyancy in this case, which is an interesting topic in itself. So you find that the cell. So what could be the reason why cells sink differently at different depths? There can be many reasons.
Rahul, question. Application really. When it says that this is the cells that are sinking, how is that determined? So
so it's a very the rough measurement is you take the cells out, you put it in a cuvette. So some cells sink to the bottom, the other rise to the top.
But you maintain, so the salinity is the same, but the pressure is different.
Yeah, pressure is different. That is true. And that is an important point in all the data I'll be presenting. I'm not maintaining the in situ pressure, which is actually a very difficult thing to do. We tried doing it, but we end up breaking our glasses. It could. So those are that's a very important point to make, in that there could be many kind of ambient field that the cell could be responding to, and the best you can do is either observe them in situ, and we don't have the facility to do that at the moment, or you can take it out and study its behavior on the ship immediately.
But the percent sinking cells, at least over here, and maybe later on, is essentially sampled out and then in the same medium, and so the physical and chemical properties are likely similar. Temperature and pressure are less controlled, but other things are the same. So for the cells that I'll be talking about, towards the end we did run experiments on going 4 atmosphere, for example, and the behavior remains the same. The sinking behavior remains the same. All right. Thank you.
It's not very sensitive to pressure. From whatever data we have collected, it's not very sensitive to pressure. So it displays. So you have to be in the upper layer of the ocean because that's what is sunlit. They need to photosynthesize, so need to be upper layer of the ocean. The bottom of the ocean, which is roughly 100 meters, is where you get nutrient-rich water. So this non-swimming dinoflagellate has to move in a controlled manner at distances as large as 100 meters, where the cells are 50 microns. So how do cells accomplish these large vertical migrations without any active swimming mechanisms? So buoyancy couples to cell cycle is what we found from. So this is the data collected in the coast of Hawaii, where if you measure the sinking dynamics, and I'll tell you how we collect this type of data set. So you are in the settling frame of the cell, and you see either sinking cells or rising cells, depending on whether they are in their vegetative phase or whether they are just newly born. Roughly, what we find, the percentages of sinking cell, correlates with, so whether the cell is sinking, so if a cell is towards later stages of its division, then it's heavier and it sinks, while a newly reproduced cell rises up, is a rough correlation that we find in these data set. So that coupling is there between cell cycle and buoyancy. We bring them to the lab and observe them under a microscope, do a long time imaging, in this case, for days. So you see individual cells. I'll leave you with this movie. So as you see, there are day-night cycles. During the night, this cytoplasm condenses to this blob, and in the day it spreads out, which increases the surface area, and there's the photosynthesis, more chloroplasts are exposed to light. But the cytoplasm is not a blob in that it's a reticulated network. And if you noticed carefully, as soon as the cell divides, it expands by 6-fold in volume. Okay. And the filaments that you're seeing are part of the cytoplasm, and everything in between is vacuolar. Okay, so conceptually, what I mean by cellular slingshots. So if you place these cells on a petri dish and focus on the bottom of the dish, just above the bottom of the dish, and observe them, what you find is as it balloon, it moves out of the focus in that it becomes less buoyant and starts rising. And those that get stuck are because of the theke. So if you look at the video, this video, there is a outside layer that encapsulates the cell, which is sticky, and the other cell ends up being enclosed in it, and that sticks to the bottom of the surface. So for those that are not able to escape, are stuck to that theke and the surface. Okay, so let's look at this in the context of sedimentation. In this video, I'm showing a single cell that as it divides, it balloons, in that it expands in volume, and due to that, it stops sinking and would start rising as the cell expands further. The way this data is collected is something that we call gravity machine, which consists of an annular fluid chamber, which is the size of like a plate that we eat in. So we suspend cells in this fluid chamber, and there is a feedback control that can rotate and translate this wheel. And depending on whether the speed with which the cell is sinking or rising, we can rotate the wheel accordingly so that the object of interest is always in front of the objective. And we can do many things with it in that we could change the pressure in the wheel. We can change the light accordingly. We can change chemical constituents to suit the ambient environment in which they are collected from. But in the data that I'll be presenting, I won't be changing any of those. I'll only be looking at sinking dynamics for short and long time scales. Okay. So as I said, if you look at these cells closely, they're formed of these reticulated networks. So cytoplasm has a certain topology to it, and we don't quite understand what's the true function of cells to have this type of topology. One simple thing you can say is it increases surface area so that the cytoplasm or the chloroplast present in the cytoplasm can absorb more light and do more photosynthesis. And in the context of expansion, it allows for cells to expand without producing more of it. You can just bring in water and then the cell can, just like opening an umbrella, for example. Okay. So in a simple picture, you start with the
yeah. Another clarification: the inflation and deflation. What's the pump? Where's the energy going?
Okay. What do you mean by which type of deflation are you talking about, Suraj?
The change in the volume needs an energy source.
Yeah.
And presumably the vacuolar space is filled with seawater or with filtered water, right?
Mm-hmm. Mm-hmm.
So where is that? What's the engine driving this?
Okay, so I'll just play this video again. So if you start with this layer, which is it's a buffer, pressure buffer, salinity buffer, and as soon as it breaks, the cell bring in pure water from which is what I'll talk about, pure water from oxide through something called aquaporins.
Okay.
So aquaporins are the ones responsible for this inflation. Deflation is not happening in these cells.
Okay.
Okay. So it's the one-time event in the cell cycle.
I see. And but
as
the day-night cycle, you had the shrinkage, right?
Yes. Okay. So that is very, yeah. So we do not so there are microtubules inside.
Okay.
And we have not looked at that problem. It's still within the tache. So there is so activity is driving it in some manner, but we have not looked at it. But the inflation is when the cell after. So if you look at post-division, the cell will pop.
Mm-hmm.
So we are looking at that event in the cell cycle.
Okay.
And that happens in the time period of 7 days, while this spreading out and condensing happens as is a circadian, like a day-night rhythm of the cell.
Okay, thank you.
Okay. So in this simple model, we can define a sedimentation time scale, which is where mu is the viscosity, G is gravity. There are three densities involved, which is seawater density, vacuolar density, and cytoplasmic density. If the cell was bringing only the seawater from outside without selective permeability, it cannot change direction as we observed. It will just slow down, but it cannot reverse the velocity vector. But so that basically says that cellular compartments are necessary for cells to, by this mechanism, for cells to change their buoyancy. And you can write down just very simple Stokes law, and you find that there is a fixed point in this space in how much it needs to expand for it to start going from sinking to rising. And we incorporate environment in this particular very simplistic dynamics through pycnocline. What that means is this density of the seawater is not constant throughout the vertical axis of the ocean. The ocean water becomes more and more saline as you go down in the ocean, and thus the density increases. And so there are three densities involved, and we define a non-dimensional environmental field in the problem. Okay. So where does this is different from the activity that Suraj just pointed out that allows the cytoplasmic content to spread out and condense. In this case, we are defining activity as just a constant parameter that drives the cell cycle and just works. And where TC is now the cell cycle time. So you think of it as a cell cycle clock, which is being driven through this, the speed of it depends on alpha. But we can actually absorb it in defining phi, which is what we do. We don't have to have this explicit parameter. And this is a very simple model in that it ignores all the nonlinearities in the feedback involved in how cells moves across in the cell cycle. And also, this is for this particular type of plankton, but I'll get to what could be the more richer features of this dynamic generically for other class of phytoplankton. Important thing to note is there is this inflation time scale. Broadly, the cell cycle can be divided into two parts. There is an inflation time scale, which is the time scale in which the cell goes from, as a newly produced cell, expands and gets a steady size. And over long times, which is order 7 days, it creates protein, acquires mass, and then divides again. And there are, okay, I'll get to that part. So there are two phases. So if you measure density of these cells at different stages, what you find is that the cell sinks towards the initial. As it balloons, it starts to rise, and slowly over long time scales, the cell accumulates mass. And since the newly produced cell has to be identical to the parent cell after a cell cycle time, the density has to be identical. So there is a periodicity in the density that has to be matched. So it's reminiscent of a boundary layer, a temporal boundary layer, where if you measure cell density as a function of phase, you have ballooning that happens at roughly 10 minutes, while there is a slow growth phase which happens in the orders of several days. In this case, it's 7 days, and all of these are evolved features of the cell which help it to survive in its environment. And all of this can be thought of. So these time scale can be thought of as an evolved characteristic of a cell. But in this case, we are trying to write down a dynamical equation for how cells move in the ocean, or this particular cell move in the ocean. So the inner region, in the experiment, we find that the cell was small to begin with, and then it expands, bringing in water that is less dense than the seawater. And its radius goes from some small number and saturates after the ballooning is complete. And this we can fit this using a tan inverse. You can fit it using multiple things, but the qualitative features of the result do not depend on the functions you use to fit these data sets. So there is a saturation that happens, and over which can be modeled as this, and you can write down the inner density based on the assumption that I mentioned before, that the cells selectively permeates less dense water from outside as it balloons. We can write down the densities exactly for that. And for outer region, basically the cell accumulates mass and becomes heavier. We approximate as a linear function, which to make progress, essentially. You could do many things here. So here we are assuming that there are no feedbacks, just to make progress, so that in the end we can add much more richness to this, to whatever we find. Okay, and the time scale are the ballooning time scale by the cell cycle time scale of order 10 power minus 3. So they're very small. The delta here is a very small quantity. If you stare at this density, you see that there is this phase divided by a small number that suggests something like a boundary layer in the problem. And we can formally define a stretched variable, which is phi over delta. And we can match the inner. And so the idea is to write down the functional form of the density throughout the cell cycle that incorporates very fast time scale of ballooning and the slow growth time scale, and then use that in the Stokes law for sedimentation and see how the cell cycle is coupled to the vertical dynamics of the cell in the ocean. Okay, if you do this asymptotic matching, it gives the intercept, and which is basically where so we wrote down the inner and the outer solution, outer behavior of the cellular density, and we want to construct a single functional form using the inner and outer. We use the standard technique, and the asymptotic matches gives us the intercept of the linear function, and constraint of periodicity gives us the slope of this function. So it determines this for us, these free parameters in the linear approximation for us. Okay, and so now you can write down the effective density value throughout the cell cycle, which formally has a boundary layer of thickness delta, which is the ratio of ballooning time scale to cell cycle time scale, which is 10 power minus 3. Okay, so from there, we write down a dynamical equation using Stokes' law by supplying the density of the cell, and beta is the environment, as I said, has the seawater density, which is sculpted in that it's stratified in the ocean, and the salinity of the seawater is different as you go down in the ocean. There's a growth dynamics captured by gamma. And if you stare at this, you see that the delta is a small number multiplies a derivative, which is a feature of a singular perturbation. So inflation can be thought of as a singular perturbation to the dynamics of the cells in the ocean. In that, if you substitute delta equal to 0, you get wrong answer. Okay. So from there, so cell divide for a. So perhaps I forgot to tell you that there is the density distribution of the cell in the ocean. You find lots of vegetative. So people have shown before that you find lots of vegetative cells. The fraction of vegetative cell is more towards the deeper ocean as compared to the surface. In that the division, more cells divide towards 100 meter depth as compared to towards the sunlit area. So we knew that. And during this ballooning time scale, the cell does execute some dynamics, but the ecologically relevant dynamics happen at long time scale, which is during the cell cycle. And so the outer solution to this dynamical equation is what we are interested in, which is how to give a projectile motion. And we can write down an effective inertial dynamics for this, which I'll come to later. So this tells us that for the cell to, so if the cell has a projectile motion, it goes up and comes down. If it does not come down to the same height over multiple cell cycle, it will keep going lower and lower in the ocean depth and thus go extinct. So that gives a criticality in the relevant density in the cell, which is cytoplasmic density and vacuolar density.
Rahul, one more quick question regarding actually the singular perturbation point that you. Given that you have, so your Stokes equation is just drag, balancing, gravity, and you don't have inertia put in, right? It's not Maxwell-Riley.
No.
You have no added mass effect.
No.
You don't have balance aspect forces and whatnot. Fall on the small time scale, can you estimate on the 10-minute time scale, is there any inertial or small Reynolds number corrections?
I see. So you're saying while the cell pops out?
Yeah. Is there a final Stokes number correction that is important because you have this short time scale?
Yeah. it's actually very slow, Suraj. The dynamic, I showed the fast forwarded movie. That's why it looks fast.
Yeah.
Otherwise, it's very gentle. So if you, for example, look at this. Oh, this video. This one. So it's very gradually inflates
Okay.
for 10 minutes.
Okay.
But so the only somewhat rapid thing is when the cell pops out of the thicket,
Yeah.
and that dynamics we do not incorporate. But that's where inertia could be important, but we have not looked at it.
Okay. I see.
So, for example, if you look at this, yeah, this dynamics is where
Second
inertia could be relevant. Yeah. But we have not incorporated it in our dynamical equation.
Yeah. Yes. Okay.
So yeah, particle inertia is not there in this equation. And we basically replace. So we start with an equation that is written in terms of z. So this is non-dimensional. Yeah, sorry. Was there a question? Okay, sorry. So this is a non-dimensional equation where we non-dimensionalize the vertical dimension by the cell size as it starts to inflate, the smallest, which is R min, and def. So we basically we map the time coordinate to the cell cycle phase, and that's how we arrive at the equation, which is closed in Zn phi. Okay, I was here. And that gives, so this also gives us a condition for escape in that if the cytoplasmic density plus the vacuolar density is not less than twice the seawater density, in the simple picture, the cells over multiple cell cycles will go extinct. And the effective inertial dynamics can be written, which gives the projectile motion. That's what the dynamics of a slingshot is, in which the ballooning sets the so in that if the cell did not have ballooning, then the cell will not reduce in density and won't change direction. So it sets the slingshot in this particular dynamics. And the effect, you can write down an effective mass, which is different from the added mass term that Suraj was mentioning. Okay. So let's say you start with a given cytoplasmic density. You fix the vacuolar density, which is set by how cells selectively permeate water from outside to inside, and ask what is the radius, how much expansion do you need for cells to go up and come down to the same height. And for small cytoplasmic density, you see that there is a finite R. So R tilde here is basically maximum ballooning radius divided by the smallest radius that it started with, n-1. And you get a finite number there, but beyond a critical cytoplasmic density, no matter how much you expand, you will not come back to the same point and go either go down, which is the gravity trap, is what we call it, which over multiple cell cycle, the whole population go lower and lower in depths, and thus won't get the sunlight that it needs to survive, unless there are some other mechanisms at play, which could be the case. Or you go over multiple cell cycles, you go up and up in the population, which is also not good, because then you lose the nutrient-rich water, which is also necessary for the cell to survive. So cell stays in this particular frame. It needs to maintain this critical parameters to survive. Okay, so in summary of this part of the result is, so plankton, by the namesake, they're thought to be as passive drifters being advected by the fluid, but they are not. even if they don't have swimming mechanism, they can actually determine their vertical position by changing their or regulating their buoyancy in multiple ways. Here we look at how the buoyancy couples to cell cycle, driven by our observation that immediately after cell division, the cell inflates 6 times its volume, dramatically changing its buoyancy. And we come up with a new, so this mechanism for motility was not known before, that cells could essentially use this inflation as a way to remain afloat. And we construct a 1D ecophysiological model where we look at the cell and its behavior in its ecological setting. In this case, it was the vertically stratified ocean. So gravity formally acts as an evolutionary selection pressure here, in that if the cell don't maintain their cytoplasmic density to below some numbers, then the whole population will go extinct in this framework. And so there are many puzzles that we still remain unsolved, that we have not incorporated in this dynamics, in that this is a boring case of a still ocean, and the ocean can have fluctuations, although it's not as turbulent as it looks. So in the skin of the ocean, the bulk of the ocean is pretty calm, but it still has some fluid dynamical fluctuations, which we can incorporate in this dynamics. And there is feedback. The cells can sense their ambient environment and respond to it. The precise way in which they do it for this particular cell, we don't know. And they can adapt. So even the environment change, they can adapt in a certain way. So they have those internal knobs. So it's not just single cells that is responsible for carbon export. It's a network of phytoplankton. And we have not incorporated that, of course. We are just looking at a single cell and seeing how it behaves and trying to get some insights into how this dynamics plays out in the ocean. To just give you another example before I move on to the next topic. So cell cycle itself is an evolved characteristic of the cell. So I talked about a particular type of dinoflagellate, but there are other type of diatoms in the ocean which are abundant, is centric diatoms. So here, think of their frustule as capsules. So as the cell divides, it inherits each cells inherit one of the frustules. One is smaller, and it synthesizes another that is even smaller than it. So one of the cell maintains a size while other reduces in size. Multiple cell cycles, half of the population will actually become smaller in size. And so size itself is not fixed. It can change as a function of cell cycle. And then cell can also exhibit like sexual reproduction. So to go back to the original size, cells actually have, so they display a sexual reproduction to execute this. So plankton in the ocean can exhibit very many things which we have not incorporated in the model, and thus many puzzles remains open, and more studies need. We need more investigation of these type of dynamics to gain better insights into how cells actually behave in their in situ environment. So just to excite you a little bit, so I showed that cells can change buoyancy only when they inflate, but I collected this video in Pacific Ocean. I'm showing a single centric diatom and some behind it can dynamically change whether it wants to sink or not, and does that very abruptly. And Suraj, in this case, inertia could be important because they change they go from not sinking to sinking pretty fast. Okay. So cells can tune their buoyancy even they don't have to wait for the whole cell cycle to regulate the buoyancy. This is what I wanted to show. So what I presented is not the whole picture of how cells behave in the ocean. And most of it, we do not know. Okay, so from there, I talked about phytoplankton, which are the primary driver of biological pump. From there, I'll go to, so these agglomerate and form marine snow. I will not talk about the dynamics of agglomeration because I do not know how it does that. Um, uh, and it's not understood very well. We look at how aggregates sink in the ocean, just to get started into gaining some insights into this dynamics. So as I said, the satellites give, like the chlorophyll gives the indirect estimate of how much primary production happens in the ocean, or how much plankton are present in the upper layer, upper surface waters. But if you hang buckets in the ocean and collect organic debris and measure how much carbon actually gets exported to the bottom, which is what carbon sequestration is, you find that as you go down, less and less of it reaches the bottom of the ocean. So this brings us to what is marine snow, first of all. So it's the food for other heterotrophic organism. In that, so it actually sustains ecosystem. It's the primary food for marine organisms, starting from tiny shrimps to bigger fish and so on. And it also sustains benthic ecosystem. When it falls in the bottom of the ocean, the creatures that live in the benthos eats these survive on these tiny aggregates. So wanted to look at how does the dynamics of this sinking microbial ecosystem plays out. And there are multiple length scales in the, as I already said, 10 power 12 orders of length scale in this problem. So what you want to do is be able to observe the sinking thing for over long times in order to get some understanding of carbon sequestration. Okay. Historically, so the William Beebe was the first one to actually, so this person in this, so a bathysphere is a steel ball, tethered to a ship. He'll go down and actually make notes of the observations that he made just with a torch. And there would be a telephone tethered to this cable, and he would just describe fascinatingly what all he's seeing in the deep part of the ocean. But it was Rachel Carson who described, called it marine snow. And I'll just read it out. When I think of the floor of the deep sea, I see always a steady and remitting downward drift of material. From above, flakes upon flake, layer upon layer, the most abundant snowfall the earth has ever seen. But it has nothing to do with snow. It is just organic aggregates. And for more detail of the history of its rich history, can refer to this very nice article by Mary Silver. So for those who have seen underwater images of the ocean, you do see these tiny specks around everything, essentially. So the idea would be, if you can take a photograph of these things somehow, from Stokes' law, can you estimate how much of it is going down? Just estimate the sinking speed and thus the flux. It turns out, if you do that, you just get different exponents in the Stokes law, in that for a given size, you can have a range of sinking speeds. So size is not a determinant of how fast the object sinks. That's one. So I started with that as a puzzle. And the idea was to, when I looked at the literature, there was much less, there was data, in situ data was very, very few, just a handful. And most of these data is actually collected by making artificial snow and then in roll. So you just bring seawater, roll it in drum, let it aggregate somehow, and then measure the sinking speed of that resultant aggregate. So in situ particles were poorly studied. There was not much data on that, especially the direct sinking dynamics. It's easy to take photographs, but to measure the sinking speed precisely in situ is a challenging problem. So what we do, if not to settle that issue, to shed some observational light on it, we go on an expedition to the Gulf of Maine. So 3 months into my postdoc, I was sent on this cruise where we started from Rhode Island and then went from Cape Cod and spent around 2 weeks in this region of the ocean. This is the East Coast. And we use sediment traps. So sediment traps are essentially, these are net traps that hang in the ocean. So we let them hang in the ocean, tethered to a buoy in the surface, and they collect material overnight or for a whole day. And then so these are freely floating, and then we latch onto it and take it out of the ocean. This is what I'm showing here. So we collect it from 80 meters deep. We let it stay there for 24 hours. So we do not collect particles that are smaller than 50 microns. So the mesh size sets the maximum size of the particle that you're collecting. And if you look at this debris, it's just so it just basically looks like this in the 6-well plate. And you can even see tiny shrimps here, these orange ellipsoids. So the marine snow is basically these greenish brown aggregate. So how do you do sedimentation assay on this? So we gently resuspend it in the gravity machine, which I described before. As the seawater, we take it from the depth at which we collected the marine snow, and it has a size range that we study is from 100 microns to 750 microns. We can't go larger, which is the size is limited by the thickness of this wheel or the annular chamber. And we don't want to deal with boundary effects in the system. We want it to be freely suspended while we are doing sedimentation studies. And Reynolds number is of the order 0.1 to 1. The maximum we found was less than 1. And thus we are, again, assuming Stokesian dynamics to be fine in this, to explain these measurements. So the dimension of the wheel, inner radius is 8.5, outer radius is this. Just like think of it as a plate in which you eat. And we suspend polystyrene beads to also measure the flow around sinking particles. And we measure 5 minutes. We take 5 minutes track. So we suspend it, find the particle, and then track it. When I say track, it basically means then this microscope is in an automated feedback where it locks the object of interest. And so you lock the object of interest, and then it rotates and translates the wheel so as to keep the object of interest always in front of the objective. Okay, but these measurements, so the goal is to be able to measure microscale flow or do microhydrodynamics in a noisy environment in that the ship is rocking and rolling. So to eliminate so there is eliminate the low frequency noise, we have a high pass filter, which is a passive mechanical gimbal. So we place our microscope setup on top. So we build this on the ship. And as you see, so when we go in the ocean, we have to clamp all our equipments because we don't want it to slide around. So we go one day before, set up our stuff before the ship even starts moving. Because once it starts moving, it's hard to actually set up anything. Then the idea would be just to take data when you're on the ship. So you have this gimbal. So the first data I collected on marine snow was very messy in that you see individual cells, you see ciliate, chain diatoms, and aggregates of different kinds, both embedded and freely suspended. And so you see many type of dynamics that happens in the first data I collected. I just picked a number density of how much suspended particles I'm adding to the seawater. And that gave rise to this complex feature where you have sea merger, foraging, and all of that. And there's also microbial activity that I'm not showing in this image, but I'll get to it. In that there are microbes that is suspended in the seawater, but also on the marine snow that decompose it over long time scales.
Rahul, just want to let you know we are about 5 minutes towards the end.
I'm close.
Just to keep track of time.
Okay. The 5 minutes towards the end, right?
Yes, yes.
Okay, cool. Okay, so we measure the sinking speed, which comes from the tracking itself, and the size. So for a given size, there is a range of sinking speed. So the idea was to measure the fluid structure interaction in the system. When you measure the flow around marine snow, you find that there are beads stuck in this invisible thing that sinks along with it. And when you plot the vertical speed, you find that the effective size of the particle is now much bigger than just what the visible images tell you. And we find that for all our samples, there was this invisible comet tail-like feature that was present in all our collected particles. So if you're thinking of fitting Stokes' law, it's clearly incorrect if you're just looking at the visible size of the particle. And there is poor correlation between visible and invisible size of the particle. So we get visible size from the visible part of the aggregate, invisible size from the fitting the ellipse around the mucus halo. But the qualitative result is that what we find is the more the mucus, the slower the particles. The presence of mucus impedes the sedimentation of the particles. And we find that by staining, specific staining, we find that these mucus is actually produced by phytoplankton. It has a biological origin. So cells in suspended release mucus so that they stick together. It provides some stability to the aggregate. But at the same time, now the aggregate is stuck with this mucus, which affects the sinking speed through fluid structure interactions. So we build a minimal model. Yeah, I won't go to. So the basic detail is that we try to lump the material features like elasticity and viscosity of the mucus halo couple it to the sedimentation dynamics. Is in that the visible particle is heavier, so as it sinks, it stretches the comet tails or mucus tails. That changes the mobility of the sinking particle. So we in a ambit of linear response, we incorporate that in a simple K-invoke type model. And to leading order in L over A naught, where A naught is the size of the undistorted mucus halo, we can write down how the distortion evolves as a function of D, which is the ratio of elasticity to weight of the particle. And there is mu by mu naught mu is the viscosity of the ambient seawater. And then there is a dynamic shape that depends on eccentricity and the non-dimensional density, rho tilde, which is similar to what we defined for the cell. So the scaling function we use is for a prolate spheroid, and long ago, I didn't go to say from scuba images that said that these particles are very much like prolate spheroid. So we stick to that approximation, and from there, so there are two equations, two unknown, in that we can directly determine sinking speed. We don't know what the elasticity is, and we don't know what the density of these particles is. So we can estimate that. So density is in the same range as the density of cells, which makes sense. And the elasticity is that of a nasal mucus. And we assume steady state in that the mucus tail has stretched and then attained a steady state. So idea is we lift the size sinking relationship to, so the way to look at the material property of marine snow contributes or modifies the sinking dynamics. And material property enters in the elasticity and the viscosity of the mucus, and the density of which is what are the constituents of these aggregates. So the idea, so we find this distribution on the sinking speed plane. The color represents this vertical sinking, non-dimensional sinking speed. If we did this measurement somewhere else, we'll find a different distribution, and the integral over it will give us the net flux, or roughly the net flux. Okay, so in summary, we conducted fluid structure interaction in field setting on marine snow and discovered an invisible mucus halo, which is a biological knob. So we don't know how climate change will affect the production of these mucus, can impede the vertical flux of carbon in the ocean. It can even stop it if so from this simple analysis, if the mucus volume exceeds beyond a certain limit, it can actually stop sinking and just stay afloat. And we don't understand how that couples, how mucus production couples to the changing environment or the environment itself. And we constructed a minimal model for Stokesian settling, and we find that the aggregate spends twice as much time in the upper 100 meters of the ocean as it would without presence of the mucus. And if it spends more time, it gives more opportunity for other critters to inhibit and decompose it. So there is a nonlinear coupling that is present, and we have not looked at it. And so there was a perspective article that showed that the presence, so this is how the climate is changing. The CO2 in the atmosphere is changing. And the difference in the atmospheric CO2 with and without mucus is over order 40, which is significant, as you can see in the scale. And our climate models currently do not incorporate that. We don't incorporate microphysics of sinking marine snow in our climate models. So microbial, I'll just leave you with this video. Well, actually, I'll leave you with one more video. But this is the video that I recently collected in. So I told you that microbes are actually an important knob in how much carbon attenuates, in that they chase it through chemotaxis. They go in these marine snow, they stay there, decompose it, and less of that. And they remineralize it in that release CO2. So the carbon that was there in the ocean that would ideally go down is now remineralized in the atmosphere. Okay. And these are dynamic trajectories, and we're still trying to make sense of how multiple different kinds of species decompose and intercept bacteria. Okay, so oceanography, to leave you with, is a classic example of cross-disciplinary science, and being on cruises with physicists, chemists, biologists, engineers, mathematicians, and it's a very fun environment in which to do science, just to advertise that more people should pursue oceanography. And leave you with this image video that I collected less than 6 months ago in the California currents. This is on Kilo Moana. So it's not just provides rich scientific setting in which to make discovery, it also is a really beautiful place where you see less pollution in the skies, at least. Thank you.
Thank you, Rahul. That was a fantastic talk. And very, very stimulating science that you shared with us. Okay, so we have some time for questions. While people should feel free to raise their hands, and we can ask questions. In the meantime, I think there's one question in the chat which we can start with. Susan Crawford asked, how big in volume are the particles you're measuring in marine snow?
So I had a plot for this. So the visible size is this. So roughly the cube of this is the volume of the visible. So an invisible size has this distribution. So it's basically the cube of the y-axis here is roughly the volume. Does that answer? The mean, the average is 200 microns of the visible part. The average is around 450 micron in size of the invisible when you incorporate the mucus tail as well.
Jan, you have your hand raised. Go ahead.
Yes, thank you. So you mentioned collisions between particles and merger. And did you observe merger of marine snow particles during this measurements?
Yeah, but I did not study it. So
yeah.
When I initially started with, So we did not know how to even, so we did not plan this assay to begin with. At least I did not have an idea of what even the kind of sample I'm gonna get. And then we started studying it, and then we decided to go from looking at this where there are the particle number density is large, and the hydrodynamic interaction between particles, differential settling can lead to more richer dynamics. But we simplify it to make progress, essentially, to dynamics where we initialize the system with less number of particles so that they don't interact with each other hydrodynamically or through other sticky interactions. But yes, to answer your question, yes, we did see merger events.
Great. I'm going to pause. I'm going to stop the recording, and then we'll continue with questions. So thank you again, Rahul.